In this video, we will discuss about the Leishman stain, what are the composition of the Leishman stain and its functions of the different components. As we know, this Leishman stain is used in staining the peripheral blood smear. So I will start with it. The Leishman stain, actually this belongs to Romanovsky's group of stain. Which also includes Giemsa stain, Wright's stain, etc. And this Leishman stain is a compound dye. Compound means it is having eosinate of methylene blue dissolved in acetone free methyl alcohol. So it is eosinate of methylene blue dissolved in acetone free methyl alcohol. So it is a compound dye. Now coming on to the composition. The Leishman stain is having basically three components, that is eosin, then there is methylene blue, then there is acetone free and water free methyl alcohol. So we will discuss each one by one. First is the eosin. Eosin is a acidic component of the dye, acidic dye, and it is negatively charged. Negatively charged. Okay. So as it is an acidic dye, it will stain the basic particles or positively charged particles. Like the granules of eosinophil and also the RBCs, and it imparts pink color. क्या देता है ये? Pink color impart करता है ना smear में. Then the next component is methylene blue. Methylene blue. This methylene blue is a basic component of the dye, basic dye or positively charged dye. It is positively charged. So as it is a basic dye, it will stain the acidic particles, acidic or negatively charged particles. Like the granules of basophil, then the nucleus of WBCs, which is having DNA, and also the cytoplasm. Cytoplasm, and it imparts blue violet color. It imparts blue violet color. And the third component is the acetone free and water free methyl alcohol. Acetone free and water free methyl alcohol. So the methyl alcohol should be acetone free and water free. Okay, what is the function of this methyl alcohol here? It has basically two functions. First is it acts as a fixative material, that is it fixes the blood smear and the blood cells to the blood slide. So the question arises, how it fixes? So what happens that this methyl alcohol it precipitates the plasma protein, and this precipitation of the plasma protein, it forms a glue like material, and this glue like material it fixes the blood smear and the blood cells to the glass slide. So what it is doing, this methyl alcohol, it acts as a fixative material and it fixes the blood smear and blood cells to the glass slide so that it is not washed away during staining procedure. And the next function of this methyl alcohol is it maintains the morphological status, morphology and chemical status of blood cells, okay, different blood cells. But this methyl alcohol should be acetone free. It should be acetone free as well as water free. First, acetone free. Why it should be acetone free? This methyl alcohol should be acetone free. What is the cause? Because if acetone is present, since what will happen that since acetone is a very strong lipid solvent, and this lipid solvent, being a lipid solvent, it dissolves the cell membrane, okay, and it causes shrinkage, crenation and destruction of cell membrane. So what it is causing this? If acetone would have been present, अगर acetone इसमें present होता तो वो क्या करता, ये जो हमारा जो cells हैं, blood cells, उसको shrinkage करता है, crenation and destruction of the cell membrane, so that the identification of the different blood cells would have been difficult, है ना, आइडेंटिफाई नहीं हो पाता. अल्टीमेटली हमारा जो स्मीयर हम बनाते हैं लिशमन स्टेन से, है ना, अल्टीमेट पर्पस होता है हमारा आइडेंटिफिकेशन ऑफ डिफरेंट ब्लड सेल्स. Okay, so it would have been difficult. Then the next point is, though, so identification would have been difficult. Then the second point is that the methyl alcohol should be water free also. But why it should be water free? If water would have been present, अगर water present होता, what would have been there? It would produce Rolex formation. Rolex formation means that is stacking up of the RBCs or piling up of the RBCs. Okay, एक जगह इकट्ठा हो जाता. Or even it may cause hemolysis also. It would have caused hemolysis. And also, if water would have been present, it would have washed away, it would wash off the blood smear from the slide. So These are the important points of Leishman stain. So anything can be asked in the Viva. Thank you very much.